Good morning, Maneda.
Good evening.
Hello. Good morning, Maneda.
Good evening.
Hello. Hello. Is it audible to you?
Yes, sir.
Good evening, one and all.
Good
My
evening,
voice
sir.
is clear.
Good evening, sir.
Hello. Is it audible to you?
Yes, it is audible. Yes, sir. We can hear you.
Right. So far, we completed in C-height plant molecular biology and biotechnology. So far, we completed block 1, that is genome organization, genome structure, and chloroplast mitochondria genomes structure and organization. Hello. Right. Okay. Is it audible to you?
Yes, sir.
Yes.
Right. Structure of genome organization and regulation of gene in eukaryote and prokaryote also completed. And in biotechnology, tissue culture process and applications, micropropagation, totipotency, these are all completed. Now we are going to discuss about in block 3, in part of plant tissue culture, the most important topic, that is protoplast culture, somatic hybridization and hybrid. Already told you about the tissue culture process. What is tissue culture? Hello?
Sir.
What is tissue culture? Simple definition, tissue culture, what we get through tissue culture. Why it is necessary? Please respond.
The cultivation of plant cell
Cultivation of plant cell.
under in vitro under aesthetic
Propagation.
conditions.
Okay.
For the growth of many plants in limited time.
In a limited time, growth of many plants through tissue culture process. Okay, that means here the culturing tissue to get a individual plants, right?
Yes, sir. In laboratory.
Actually, in what way we get individual plants in general? How the plants are obtained? I'm asking about in general conventional method.
Sir, in general, we get plants through seeds, sir.
Seed germination.
Right, how the seeds are formed?
Fertilization.
Production of a plant from the
Pollination.
production of a
Pollination.
plant from a single cell.
Pollination,
Not pollination, fertilization.
germination,
Right, what is fertilization?
fertilization. Yeah.
The fusion of male gamete and female gamete, they will produce
Fusion of
zygote.
male and female gametes.
Right, if the fusion takes place between male and female gamete, as a
Female
result,
gametes.
formation of zygote, which right. As a result, it developed
Yes,
into
sir.
diploid zygote and further developed into diploid embryo, then seed and developed a plant. It is a time-taking process.
Plant.
And
Yes.
we do not
Yes, sir.
we do not get the desirable character. What we get desirable character, we don't get through the conventional method.
Yes, sir.
But in tissue culture process, we selected the explant from the desired plant, and this eggplant can grow as a new individual plant through the in vitro process by tissue culture techniques. In the same way, here another tissue culture method also there, that is protoplast culture. What we learn in tissue culture, the culturing the tissue or explant. The explant may be the root, shoot, leaf, flower, pollen grains, whatever it may be, they are considered as the explant. In the same way, like root shoot, now we're going to use about the explant from the protoplast. Now the protoplast is the explant. Right? What is a protoplast? What is a protoplast?
Cytoplasm and the plasma membrane. Together they call protoplasm.
First of all, what are the cell components?
Cell membrane and
Cell
protoplasm.
components.
All the cell
Just
cell
wait.
organ, cell wall, plasma membrane, cytoplasm.
Just wait one minute. Let me show diagram. Right? Are you able to see this diagram?
Yes, sir.
Hello?
Yes, sir.
Right. So this is the plant cell structure. Right. This is the plant cell structure. In plant cell, so the outer membrane, the outer layer called as this outer one, the green colored outer layer. This one color.
Plasma membrane.
Can you visible
Cell wall?
marking?
Yes, sir.
This is the
Plasma
cell wall,
membrane.
right? This is the cell wall, not a
Yes,
plasma
sir.
membrane.
No,
Don't
no.
get
Yes,
confused
sir.
between the plasma membrane cell wall. The outer layer is considered
Outer
as the cell
layer.
wall. Right.
Yes, sir.
If we remove this, the outer layer, that means cell wall, the rest of the content of the cell called as
Protoplast.
So
Protoplasm.
this entire called as protoplast. Right? Our
Yes,
topic
sir.
is protoplast culture. Right?
Yes.
Protoplast culture.
Culture.
Right. So first of all, just wait. The protoplast culture the protoplast culture technology developed by Carlson, Power and Cocking in 1970. And this, so for protoplast culture, we need to separate cell wall, right? Hello?
Yes, sir.
Yes, sir.
We need to separate the cell wall. This cell wall we should remove. Then only the protoplast removed out. Now it is convenient to culturing easily. Right now, how can we separate the cell wall from the plant cell? So there are two methods to separate the cell wall from the plant cell. One is mechanical method. Another one is enzymatic method. So first of all, be careful. The first you develop the concept. What is the protoplast? Protoplast, except the cell wall, the entire content of the cell called as protoplast. So for the culturing of the protoplast, we should separate the cell wall because the cell wall outer protective layer. Only we separate the cell wall, it can easily culturing, right? So for the separation of the protoplast, there are the two methods. One is mechanical method. Another one is enzymatic method. What are the two? Mechanical method and enzymatic method. Please, all of you request, please take pen and paper. That means one rough notes.
Yes,
please
sir.
note down the points.
Okay, sir.
Yes, sir.
Right. So after protoplast definition, now that means protoplast culturing. Now we have to separate protoplast from the plant cell. For this, there are two important methods. One is mechanical method and second one enzymatic method, right? Let's see the mechanical method. Through mechanical method, how we separate the protoplast from the plant cell. Let's see. Mechanical method, just wait. Through mechanical method, right. Are you visible? Hello? Is this visible to you?
Yes, sir.
Visible, sir.
Yes, sir.
Right. So we can see here clearly. So this is the plant cell, right? Plant. for plant cell, the outer, this wall layer, this is the called plant cell wall. So this is the round shaped structure. This is the protoplast.
Protoplast.
So we need to separate the protoplast
Sir,
first.
it is not showing. It's showing.
Okay.
We
Right.
can see.
Yes, sir. Yes.
Right. So through mechanical method, we can use sharp edged knife for releasing the protoplast. So before that actually the plant cell in general fully turgid. So we need to plasmolyze it. The plasmolyzed cell. What is plasmolysis? What is plasmolysis?
Plasma membrane, sir.
Right. Actually, are you able to see the diagram?
Yes, sir.
Yes, sir. It's visible, sir.
Right. So the red color line cell wall, right?
Yes, sir.
Yes.
Are you able to see the yellow color line?
Yes, sir.
Yes, sir.
Right. So this yellow color line, plasma membrane, with its inner portion, protoplast. That means the plasma membrane and protoplasm together called as a protoplast. So this from the yellow boundary, entire yellow boundary, and the inner portion also completely called as protoplast. Actually, this is the full-filled cell. Can you cut the cell wall in this type of cell? Is it possible to you? Hello?
No. No, sir.
Because
No, sir.
actually, for our convenience, here I leave the small gaps. Actually, the full-filled cell, there is no gap between cell wall and plasma membrane. No gap. So we unable to cut the cell wall. That's why what we do?
Enzymatic action.
No. In mechanical method, what we do for the separation of the plasma membrane? Actually, in
We have
general,
to use
Plasmolysis.
sharp knife
Right,
to cut the cell wall.
right. First listen carefully. In general, what I'm saying here, the plant cell wall and plasma membrane, these are firmly attached to each other, so that we cannot cut. Cannot cut.
Yes, sir.
Only the plasmolyzed cell, we cut the cell wall. How it is get plasmolyzed first? This full-filled cell, how it get plasmolyzed? What is plasmolysis?
Breakdown.
What is means lysis,
It's
right?
a contraction of a protoplast.
Right, shrinking of protoplast, right? So how it
Yes,
is
sir.
shrinking?
Loss of water. There will be loss of water.
Sir, loss
when-
of water in protoplasm.
When the loss of water from the cell,
The transpiration
when it
process.
happen?
Dehydration.
Then, sir, when the
Transpiration.
concentration around the cell is more,
Right.
the cell,
So right. That is called the more concentration. That is called when the cell, the full-filled cell, placed in hypertonic solution.
Hypertonic.
Right. That means high concentration solution. What happened here? The cell content water released out. So as a result, after plasmolysis, that means when we placed in hypertonic solution, immediately the cell wall as it is, but the plasma membrane with protoplast shrinken like this. Okay? Are you able to see?
Yes, sir.
Right. So this is called, this is the now the protoplast. Now there are the small gap between the cell wall and protoplast, right? Hello?
Yes.
Yes,
Right.
sir.
Yes, sir.
Now see the third diagram first. Just wait. First, second, and third diagram. As it formation gap between protoplast and cell wall, now we can easily cut with help of knife. Sharp edged knife, with help of sharp edged knife. Right? Now,
Yes.
what happened after removing the cell wall? After removing the cell wall, what happened? This protoplast separated. Now we get protoplast only, right? This is the separation of protoplast. But here, the removing of the cell wall from the cell is difficult task, isn't it? Hello?
Yes, sir.
Removing the cell wall from the cell is the difficult task,
Yes, sir.
and
Yes, sir.
care must be taken and not to damage the protoplast. Right? We take the necessary action for this method. In mechanical isolation, and also in mechanical isolation, it is very difficult to achieve. As a result, low yields and poor quality of protoplast we get, because there is a easy chance to, while we using the knife, this knife, there is chance to touch the protoplast and damage the protoplast also. And for this, skilled person we required. So mechanical isolation of protoplast from higher plants, first pioneered by Klerker in 1892. Klerker. Write down Klerker. Klerker in 1892. 1892. Right? Is it clear? First mechanical method, first process.
Yes.
But is it easy or hard? Is it convenient to us?
Yes, sir.
Is it convenient? Mechanical method is convenient or not?
Yes, sir.
Not convenient.
No.
Not convenient. It is more risk process, and
Yes.
it can damage easily. That's why we choose the another method, enzymatic method. Right?
Yes, sir.
First mechanical clear.
Yes, sir.
Right now, second method, enzymatic. The first enzymatic method discovered by E.C. Cocking in 1960. And he gave out first outlined feasibility of enzymatic digestion. Who is E.C.? Who is he?
Enzymatic digestion.
E.C. Cocking.
Sir, spelling, sir, spelling.
Just wait. Just wait. Right, please keep quiet. Hello. Thank you. Durga Srinu. Thank you. Right. Second method. Are you able to see that board?
Yes, sir.
Yes, sir.
Yes, sir.
Right. There is no proper diagram for enzymatic method. That's why I had to use this whiteboard. So first, this is discovered by E.C. Cocking. Please in silent. Cocking in 1960. So first of all, what is enzymatic method in general? What we use in enzymatic method? Hello?
Some enzymes were used to break down the cell wall.
So enzymes use. Which enzyme use? Before that, you should know about the cell wall. Chemical nature of the plant cell. Chemical nature of the plant cell wall. Do you know chemical nature, chemical composition of plant cell wall?
Cellulose.
Right, cellulose.
It contains
and
cellulose.
cellulose common, and apart from the cellulose,
Pectin.
pectin.
Pectin, sir. Pectin.
Right, apart from the cellulose, pectin also present. So which enzyme involved in this? So here just listen carefully, just simple. So here the substrate is cellulase, cellulose and pectin. So because of this cellulose and pectin, the plant cell wall have the rigidity, right?
Yes,
And tough in nature. If we make it soft, the cell wall, what will happen? The cell wall completely dissolved. As a result, what will come out?
Protoplast.
Right, protoplast come out. So that was in this process. We can use enzymes. That enzyme should be.
Sir, repeat.
Hello.
So once more.
So first of all, we should learn the chemical composition of the cell wall, so that we dissolve the cell wall. What is the enzyme? What is the enzyme importance? What is the enzyme function? Simple. What are the enzymes? What are the main function of the enzymes?
It will enhance the rate of the reaction.
That means digestive. Simple digestive breaks.
Break down the
dissolve the substrate.
Yes.
So the chemical cell wall chemical is pectin, cellulose, and pectin. Because of this chemical, mainly the cell wall have the rigidity and stuff. If we use opposite to the substrate and this substrate, enzyme always end with ase. The enzyme which act on cellulose called as cellulase. Cellulase, right?
Yes, sir.
And the same way on the pectin. Pectin is right. Pectinase. Pectinase. Let me clear. Pectinase. Pectinase, right?
Yes,
so with help of these two chemicals, pectin, pectinase, and cellulase. Pectin and cellulose, the both are dissolved. As a result,
protoplast will come out.
Right, protoplast will come out. Right. So most workers use this one step of method because it is less time consuming. When compared with mechanical high risk and time taking, should be skillful
And difficult.
Right. And another default of the mechanical isolation, another default is it can easily infected with microbes. Mechanical method, because the mechanical method we use the knife, that instrument. Maybe instrument already affected with some microbes. That's why easily the protoplast infected. But in this method reduces the chances of microbial contamination. Microbial contamination by enzymatic method. So this enzymatic method more comfortable than mechanical method. In general, the technique for protoplast isolation involves the surface sterilization. First of all, before protoplast separation or protoplast isolation by any method, either mechanical or enzymatic, we should sterilize the material so that not to infect. For this, the material fully expanded. That means first, for example, if we take the leaf material, we separated the leaf peeling carefully with help of the forceps, and the peeled out this, and later the put inside the hypertonic solution. What happens if we put inside the hypertonic solution? Automatically the leaf peeling cells of water lost. As a result, protoplast sinken. Now we can easily digest the cell wall. In two processes, in mechanical method or enzymatic method or any other method. First step, we should sterilize the plant material. After separate the protoplast should be inside the hypertonic solution. In third step, what will happen? Due to hypertonic solution reaction, the water loss from the protoplast. As a result, the plant cell wall protoplast shrinken. Now we can easily separate the cell wall by mechanical method or enzymatic method. Right. So this is enzymatic method. Any doubt in this?
No.
Hello?
Clear, sir.
Is it clear?
No, sir.
Tell me. Any doubt? Hello? Someone
No,
Sir, it's
told
clear.
no.
Sir.
Right.
it's clear. Clear.
Okay. Right. Okay, anyway, by two method. Two method means by
Mechanical and enzymatic.
protoplast, mechanical method and enzymatic method, we separated protoplast. Right? So we can now we should check the viability of the protoplast. What we get the protoplast by mechanical or enzymatic. After getting the protoplast, we should check once whether it is viable or non-viable. That means whether it is good or spoiled or damaged. So there are, we can check by three methods. First of all, the observation of cyclosis or cytoplasmic streaming. Like cyclosis, cycloplasmic streaming. The second one, oxygen uptake by protoplast, which measured by oxygen electrodes. And third one, photosynthetic activity. In this, the most general measurement, the cyclosis method. What is cyclosis? What is cyclosis? What we said, just wait. So, after get the protoplast, just wait. After we get protoplast, have we got the protoplast?
Enzymatic or
enzymatic or mechanical, but most preferable is enzymatic, right?
enzymatic.
Right. So anyway, somehow we get the protoplast. But whether we should check whether it is viable or non-viable. The checking by, first of all, observation of cyclosis. Cyclosis simply the active movement or streaming, active movement of streaming of the protoplast. If the protoplast, easily moves from one place to another place, that means, for example, this is the cell, this is the, just wait, the red color protoplast, the separated protoplast, not stable. It can direct like this, follow arrow mark, like this direction. So this is called cyclosis. If you observe this cyclosis process, it is viable protoplast. It not show the cyclosis process, it is considered as a non-viable. Non-viable means which is not used for culture.
No movement.
Not useful culture. That means it may be infected with microbes or it may be damaged while we separated. So another one is with help of oxygen electrode method, we estimate the oxygen uptake. If it is observe the oxygen, it would be considered as a viable. Unable to observe the oxygen, we consider as a non-viable. right? So last one is photosynthetic activity. How can we measure the photosynthetic activity? That means in protoplast, plastids are there. The plastids actively involved in photosynthesis to prepare food material. This is carried out only in viable protoplast. What happened in non-viable protoplast? Is it possible to photosynthetic activity, photosynthesis process?
No.
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 So mainly these three methods, we will have to check whether it is viable or non-viable. So anyway, after we check, what will we do next? First, selected explant in the form of protoplast. After that, we separated the protoplast in second step. After that, we check whether it is viable or non-viable. So three,
Viable or non-viable.
right? This three clear. Now, what is next step? What will we do?
Culture.
Right. Culture of protoplast. Culture of protoplast. Simple. For culture of protoplast,
Sir, sorry for interrupting, sir. Sir, please
okay.
repeat the second and third method, no, sir.
What is first method first?
First one is cyclosis, no, sorry. The protoplast, it shows
Right.
cyclic movement.
Okay. You are asking about viable process. First one, cyclosis. Second
sir.
one, right. Let me write. Third one, oxygen uptake. Oxygen uptake by protoplast. Oxygen uptake. We measured this oxygen uptake by oxygen electrodes. By electrodes, right? Next one, photosynthetic. Yes, means photosynthesis activity. Now clear? Hello?
Sir.
Are you clear?
Yes, sir.
Yes, sir. So,
Yes, sir.
means, sir, how we will define it by oxygen electrodes, sir? It is viable or not? I didn't understand that, sir.
Based on oxygen uptake, mean observation percentage. Observe, that means actually in electrode method,
Sir.
for example, this is the one container. Just wait. Right. This is, let me change color. This is protoplast. Electrodes. Some electrodes. These electrodes out oxygen percentage. Oxygen percentage in protoplast. For example, just wait. This is the protoplast. In protoplast, oxygen atoms. Oxygen atoms number. These oxygen atoms actually closely attached with these electrodes. Due to the activity, negative activity, these are closely attached with these electrodes. Through this process, we selected this that means the protoplast. Through the protoplast, oxygen molecules, whether present or absent. There are no oxygen molecule in the protoplast. When selected protoplast, there are no oxygen molecules around the electrodes. Based on this, we confirmed whether it is viable or non-viable. That means electrolytical charge by negative positive charge. Based on negative positive charge, we can estimate the oxygen percentage present or absent. Is it clear?
Yes, sir.
Right. And next one, common photosynthetic method. But actually, we do not follow the second and third.
Sir.
Because the cyclosis we can easily identify. We can see in microscope the movement of protoplast. So this is the easy process to estimate viable of protoplast, non-viable of protoplast. These two very difficult electrodes usage and also time taking also. And in third method, how can you define the photosynthetic end product? Whether end product increase or decrease? Of course, photosynthetic activity, based on photosynthetic activity, we confirmed the viability of the protoplast. But it is difficult to find out the end product of carbohydrates. Right? So among these three, the most easy understanding cyclosis process. Simple. Is it clear?
Yes,
Hello?
sir.
Yes, sir.
Can I move forward?
Yes, sir. Yes, sir. Thank you, sir. Thank you so much, sir.
Right. Right. Culture of protoplast. Culture of protoplast. In protoplast culture, right. Protoplast culture. So the separated viable protoplast, first step, expanded on agar medium. Expanded on agar medium. Agar is the act as a substrate. For culturing, need a substrate. We require a substrate, right? Hello?
Yes,
Hello?
sir.
Yes, sir.
Yes, sir.
Right.
Yes, sir.
So this, for example, this is the agar medium. The red color, protoplast. Right? First,
Yes.
this is isolated protoplast. Take a surface of the agar medium. The agar medium already have this agar medium already have some nutrients. Nutrient means vitamins, hormones, hormones, and other nutritions. That means elements, ferrous, carbon, magnesium. Like this, other nutrients also present inside the agar. And the temperature should not exceed 45 degrees centigrade. Please be silent. Hello. Ramadevi. Hello. Hello, who is Rama Devi?
సార్ హోస్ట్ మ్యూట్ చేయొచ్చు పార్టిసిపెంట్స్ ని.
Option okay. Right. In agar-agar medium, already filled with nutrients and the temperature should not exceed 45 degrees. So this is the first step. And second step, after observing these nutrients and proper temperature, second step, incubation in thin layer of liquid medium. Second step, right. So first agar-agar medium we utilized. Next one, incubation. What is incubation? Incubation, the protoplast. Incubation protoplast or thin layer medium. Protoplast with agar medium. Protoplast with agar medium. So incubating protoplast in thin layer. Actually, thin layer means here the protoplast applied as a layer on agar medium. That means agar nutrient medium inside the petri dish. Already told you. For example, this is the petri dish layer. Second yellow layer, agar-agar. Spread agar-agar. Agar-agar. On the agar-agar, what we apply? Agar-agar nutrient medium, what we applied?
Protoplast.
Right, protoplast. Now covered with petri dish. After that, this petri dish inverted, turns into upside down. Right? That means upside down into the another petri dish, another petri plate. After that, finally sealed with paraffin. Finally sealed with paraffin wax. Now this culture, the maintained in low temperature, dark condition at 25 degree to 28 degrees centigrade. Right? In first step, not exceed 45, but during incubation period, it should be 25 to 28 degrees centigrade. After that, we separate, we take the small micro drops. In third step, after incubation, from this protoplast, we take small micro drops. This small micro drops, take another petri dish with already nutrient medium. These micro drops take into the another micro drops. That means, for example, I take 3 microdrops. These three microdrops fully expanded and fully developed as a callus, later developed into individual plant through the culturing process. I will tell you later. So anyway, after incubation period, take some microdrops from the incubate culture, right? The culture is then maintained in low light dark at 25 to same temperature should be maintained, 25 degrees centigrade to 28 degrees centigrade, right? Fourth step, plating protoplast and solidified agar. Plating protoplast and solidified agar. That means actually this microdrop separated and shifted into another petri dish, right? This another petri dish already with agar medium, agar nutrient medium. After that, cover with plate. After with cover with plate and let them incubate. That means let them absorb the more nutrition from the agar medium for the regeneration of the protoplast. This regeneration carried out the same low light dark conditions 25 to 20. This is the optimum temperature. Remember, the optimum temperature for the culture of the protoplast 25 to 28 degrees centigrade. This centigrade, the protoplast readily regenerate within the 2 to 4 days. Undergoes the some divisions. Due to these divisions, these microscopic colonies are formed. These microscopic colonies are transformed into normal tissue culture media. Right? So in fourth step, what will happen? And finally, from these micro drops, later these micro drops called as a colonies, like this. Colonies are formed. After formation these colonies, now these colonies transferred into the in vitro media. Generally, what is the general medium in tissue culture? What is the most preferable media? Hello?
Growth medium, sir.
What media? Mursuges Krug media. Mursuges Krug MS media.
M.Sc.
And another media B5 media. So anyway, the rest of the process, I think you know. Already I explained about it. That means after transfer into the MS media,
Sterilization,
the MS media should be sterilized, right?
inoculation.
And should be inoculation, incubated after 3 to 4 weeks. What will happen? What will produce from the protoplast colonies? Finally, after observing cytokinins and auxins, nutrients, other nutrients, vitamins.
Transferred into polythene covers.
No. Before transferred, what are the produced from this protoplast colonies?
Tissues, sir. The preferred
Yeah.
tissues.
Colonies nothing but tissues.
Yes.
Okay.
Actually, we selected the tissues.
Sir.
These tissues transferred into the MS media or BF media, a B5 media.
Sir.
But during this protoplast culture, what happened?
Tissues develop into plant parts, sir. Which
Plantlets, small plantlets are generated inside
yes,
the
sir.
in vitro media. The small plantlets now finally transferred into the polythene bag and
Greenhouse.
protect in greenhouse 2 to 3 weeks. After that, transferred into the outer atmosphere. So this is the process, right? Is it clear?
Yes, sir.
Right? Remember these 4 steps first. Agar selected in general language, first selected agar media suspended as a thin liquid layer. Suspended the protoplast as a liquid layer. right? Incubation. Next one, next third step, culture. Micro drops are separated from the incubation and shifted into another petri dish and sealed with the petri dish should be sealed. Next, this product during after incubation period, the small colonies are produced. Small colonies are produced. Colonies, this produced colonies shifted to the media, MS media or B5 media. The further process, already it is explained. Further process was explained already in tissue culture techniques, right?
Yes,
Hello?
sir.
Yes,
Yes,
sir.
yes, sir.
Right. Now somatic hybridization. So this process, what we all process called as hybridization fusion of protoplast. So this fusion, the protoplast fusion carried out by chemical method or electrical. These two are main chemical method, electronic method. Let's see the first chemical methods. Just wait. Right. See diagram. Hello.
Yes, sir. It is visible.
Somatic. Why we can't somatic hybridization first? What is hybridization? What is hybridization? Hybrid formation by fusion of male and female gamete. Here the somatic means involved somatic vegetative cell. That means we selected the protoplast from the cell, vegetative cell, right? For fusion. Remember, this is the leaf plants. This is another plant leaf. So this is this has some desirable character. This also have some desirable character. That's why we selected two closely related species. Remember, the protoplast fusion and somatic hybridization provide alternative in the way for transfer of traits between distantly related species. This is takes place only between related species, right? So selected related species, that means related desired character species. From this we separate the mesophyll cell. This is the mesophyll cell and this is the mesophyll cells also. After that, by isolate methods. What are those two methods? Hello?
Mechanical and enzymatic method, sir.
Right. Anyway, mechanical method, enzymatic method also, protoplast also separate. Now the separated protoplast, after make sure whether it is viable or non-viable. Now fusion takes place between this. Fusion takes place. From the fusion, we know some of the methods. That means we prepared some agar medium, agar nutrient medium. We should be incubated. We separated some micro drops from the medium, and after incubation, separate the culture colonies and transferred into the medium. But fusion in agar-agar, what will happen when we apply agar medium? For example, this is petri dish, right? Hello, red color, clear? This red color clear, right? agar media, yellow color protoplast, and another right, green color also protoplast. Green color also protoplast. This is yellow, one plant protoplast. This another protoplast. So this two should be fused. This should be will fuse together. The fusion of these two protoplasts, we required some chemicals are required from electrodes, right? First, what are the chemicals involved to fusion of protoplast? Let's see. First, chemical fusion. In chemical fusion, first this is discovered by and Michelak, Cow and Michelak in 1974. In chemical method, mainly used the most useful chemical is PEG chemical. PEG, that is called PEG. P for poly, E for ethylene glycol. Polyethylene glycol. The polyethylene glycol has been identified as a potent fusogen. Potent fusogen means which chemical used for fusion of protoplast. That is called fusogen. It is a high molecular weight and water soluble polymer. Why we use only PEG and it is effective because of one reason, high molecular weight and water soluble polymer. Generally, the PEG solution is used for protoplast fusion. The polymer binds with the lipid membrane of the cells and thus induce the fusion. Right? Here, what are present in protoplast? Water. The PEG, that is polyethylene glycol, easily dissolved in water. As a result, these two protoplasts fuse together, easily fused together. So this is the one chemical, and another chemical is, this is separated protoplast, this separated protoplast, another protoplast, after fusion. These two separated protoplast fusion by PEG chemical, polyethylene glycol chemical, right? Another chemical, high concentration of calcium ions also. Let me change color. So another chemical. So this is one method of chemical. Another method, high concentration of calcium. High concentration of calcium ions. High concentration of calcium ions at high pH between, apart from the calcium ion, the pH should be maintained 8 to 10, should be maintained 8 to 10, and temperature should be maintained up to 37 degrees centigrade. So in this presence of high pH and 37 degrees centigrade, preferable for protoplast fusion with help of calcium ions. Another chemicals, so PEG 1, next calcium. Another one is dextran, polyvinyl alcohol. Let me change color here. Dextran and polyvinyl alcohol. Polyvinyl alcohol. And synthetic phospholipids are also used as a fusogen. So in chemical fusion, we should use PEG chemical, high concentration of calcium ions,
calcium ions,
dextran, polyvinyl alcohol,
dextran, polyvinyl alcohol.
synthetic phospholipids. Right? This is chemical. Is it clear?
So lipids. Okay, sir.
Are you in doubt?
No,
Say
sir.
confident. Are you clear or not?
Sir, at what
sir,
stage
clear.
we add, sir, these chemicals?
What?
Sir, at what
What
stage we
chemical?
add these chemicals? Means during the incubation or
No, before incubation. In first step.
before culturing,
When you right. When you applied the protoplast,
sir.
two different protoplast
Sir.
on the agar nutrient medium, then only you had the chemicals.
Yes, sir.
Right? So that they can easily fuse. And they can easily fused due to fusion, the both protoplast nucleus fusion.
After this fusion only we go with these steps, sir. Means the culturing of the cells.
Yes. Okay.
Okay, sir.
Right. And next step. Is it clear? First chemical.
Yes, sir.
What is the next one? Electrical fusion. Electrical fusion is a more reproducible method of damaging chemical due to the chemical composition. For example, in chemical composition, if we use less chemical or high chemical, it may chance to damage the protoplast. But in electrical method, less damaging than chemical fusion, but requires expensive equipment for the electric fusion. Let's see electric fusion. Just wait. Let's see electric fusion. Electric, where it is. Able to see diagram?
Yes, sir.
Right? So in electrofusion, the alternate generation of AC field and DC pulse are two steps required for electrofusion. That means AC current, DC current, that is the different to explain because it is the part of the physics. So anyway, this electrode AC current with AC and DC current applied to this liquid. Already this liquid added the protoplast. They are all the protoplast. Due to electrode charge, this protoplast come closely and fused each other. Come closely. Fusion of protoplast. Is it clear?
Yes, sir.
Hello? So simple in electrode method. Speak out.
Yes, sir.
Clear?
Yes.
In electrode method, simple electrode applied with AC current and DC pulses. Mechanism, the already existed protoplast each other. What you did? Here? Hello?
Yes, sir.
Yes, sir. Clear, sir.
Right. So, okay, by any method or chemical or enzymatic method, any method, chemical method. Homokaryons and heterokaryons are formed. What is homokaryon?
Same type of nutrient. Present for
Same. That means within the plant. This is the one plant. A plant. From the A plant, you selected the two type of protoplast. From the somatic cell, from the A plant only two type of protoplast selected. Now, as a result, formation after fusion of nucleus, that is called homokaryon. In one A plant, another in B plant, A plant protoplast separated and B plant protoplast separated. As a result, fusion takes place. Now it is called as a heterokaryon. And remember one thing, dikaryotic condition also formed. Remember the dikaryotic condition also formed. What is dikaryotic condition? Hello?
2N.
In sometimes what will happen, this is the one protoplast, this is another protoplast. Actually in both protoplast, nucleus present. In general, these two nuclei fused, but sometimes these are not fused, but lie within the protoplast fusion. That means only two, this red color protoplast, yellow color protoplast, red color nucleus, blue color nucleus. Not fusion, two separate nuclei present within the closed cytoplasm. Now here the cytoplasm fused, but not nucleus separate.
Nucleus.
In this time, the dikaryotic condition also formed. Dikaryotic condition. Is it clear?
Yes, sir.
Carlson. First Carlson. Just wait. Carlson in 1973 first reported somatic hybrid. First reported somatic hybrid by fusing a protoplast in Nicotiana. Protoplast culture, somatic hybridization, protoplast culture, first developed by Carlson. The plant name Nicotiana. Plant name Nicotiana, right?
Nicotiana.
Clear?
Yes, sir.
In 1978, another scientist, the name Melchers, the same protoplast fusion takes place between the, but the first time in Nicotiana, but second, Milcher, the protoplast culture between tomato plant plus potato plant. Tomato and potato. The fusion of cells protoplast of tomato and potato. As a result, named as a tomofel. Important, tomofel. It is competitively very important. Tomofel obtained by protoplast culture between tomato and potato. And obtained a new plant that is called tomofel, or another name carmate. Carmate. This is proto culture. And another one is protoplast culture and somatic hybridization. And next one is hybrid. Take diagram. Hybrid. Somatic hybridization very important for long question. Remember, somatic hybridization very important for long question and hybrid for short question. Hybrid formation. Simple in hybrid, the fusion of cytoplasm only. Remember it, fusion of cytoplasm. What is different between protoplast and cytoplasm? Hello?
Without plasma membrane is called as cytoplasm.
Without plasma membrane?
Yes, sir.
Mhm.
All the cell organelles and the cell sap is called as cytoplasm.
For example, you separated the protoplast here, nucleus.
Yes, sir. Yes, sir.
Here protoplast without nucleus.
Protoplast.
With no nucleus, without nucleus, this is called as a cytoplasm. Without nucleus, called as cytoplasm. Now this nucleus, protoplasm, without nucleus, cytoplasm, fused. As a result, one type of nucleus present inside. Only fusion takes place between the two cytoplasm. That's why it is called as a hybrid. That's why it is called as hybrid. Right? Hello? Hello?
Sir,
Yes,
cells
sir.
don't have
సో
nucleus.
టూ ప్రోటోప్లాస్ట్ ఒక దాంట్లో న్యూక్లియస్ ఉంటుంది ఇంకో దాంట్లో న్యూక్లియస్ ఉండదు అదే కదా సార్
Nucleus may be present if it is may be absent if it is present that nucleus inactive condition. That is called as a male sterility. That is called as a sterility. Sterility nucleus may be present. That means not involved for fusion. Okay?
ఓకే సార్
Right. So anyway, protoplast culture, there are some applications, some important. What are the important? Let's see. Disease resistant plant. The same application. Well, that means tissue culture technique application. What are the tissue culture application? Disease resistance, environment tolerance, quality characters, high yielding plant production. They are all the same applications carry forward here also. The first one is disease resistant. Disease resistance. Environment tolerance. Third one, quality character. Write down, these are all not given in our book. Please write down, these are all. Quality characters.
ఓకే సార్
Quality characters. Uptake. Foreign material. uptake foreign material, and it is gene transfer. Gene transfer. Specific or intragenic hybrids created. Many disease resistance are there. Example, tobacco mosaic virus. Resistance. Next, environment tolerance. What is environment tolerance? The gene responsible tolerance of environmental issues, frost, salt, high temperature also, and resistance to high temperature also environment issue. So these are all carried out by environment tolerance through we get through the protoplast culture. And quality characters. Somatic hybrids for the production of high nicotine content. That is the quality. For example, a tobacco plant have the high nicotine content. We selected as a desirable plant and we obtained the protoplast, that plant only. And culturing from this plant, protoplast, as a result, we get desirable character plant. Because the tobacco plant have the nicotine. That's why get quality character. Second, uptake of foreign material. Actually, the protoplast, no protection layer, right? Hello?
No protection layer.
No.
Right. Yes, being naked in nature, the isolated plant protoplast are considered to be very suitable for genetic and cytoplasmic modification experiment, because they are these are already exposed one, no protection layer. So we can separate nucleus easily, we can separate DNA easily. That's why we uptake the foreign material. The scientists have reported the unique property of uptaking of isolated nuclei DNA or chromosome chloroplast. For example, nitrogen fixing bacteria and even virus particles may be plant protoplasts. For example, nitrogen fixing bacteria. A nitrogen fixing bacteria, this is the, just wait, bacterium. What is the nitrogen fixing bacteria? Hello?
Rhizobium.
Right, rhizobium bacteria. From the rhizobium bacteria, that means dissolve the cell wall, next the rest of the cytoplasm, we separated the DNA. This DNA mixture in another plant protoplast. Due to the fusion of this DNA and plant protoplast, so what will happen after obtaining new individual plants? Nature of rhizobium bacteria DNA. Rhizobium bacteria, actually any character under control of, we have some specific character.
Gene.
So rhizobium bacteria specific
yes.
character is fixation of atmospheric nitrogen. What if we introduced this rhizobium bacteria into the desired plant?
So it also fixes nitrogen.
Right, that's it. And gene transfer. Last one, gene transfer. Gene transfer also, this is one type of uptake foreign material. The protoplast are the ideal material for the gene transfer. Actually, in general, what is the natural vector for gene transfer? Hello?
Plasmid, sir.
Right, plasmid. Right. This plasmid is the part of which organism?
Bacteria.
Right. Mainly the agrobacterium. So instead of agrobacterium, we can directly use the protoplast as the vector. Right. So direct gene transfer by using isolated protoplast is an alternative to the use of agrobacterium. This is the natural. Agrobacterium is natural. But instead of agrobacterium, we can use protoplast for the gene transfer. Okay.
Okay.
Actually, this agrobacterium considered as nature's genetic engineer. That is common, naturally formed, but protoplast man-made. So we can use instead of nature genetic engineering,
Agrobacteria.
so-called nature genetic engineer agrobacterium, we can use protoplast. Right? Clear?
Okay.
Applications of
Applications of
a protoplast culture, somatic hybridization. Okay.
Hybrids
Actually,
somatic hybridization.
సార్ ఇది ఒక సెల్ లోనే ఒక న్యూక్లియస్ ఉంది, ఇంకో సెల్ లో ఓన్లీ మనకి సైటోప్లాజం తీసుకున్నాం. అలాంటప్పుడు ఫ్యూజన్ అయినప్పుడు సింగిల్ న్యూక్లియస్ మాత్రమే ఉంటుంది ఆ సెల్ లో.
అవును.
ఫర్దర్ గా ఎనీ ఫ్యూజన్ కి పార్టిసిపేషన్ ఉండదు కదా సార్?
లేదు. సో
అలాంటప్పుడు హైబ్రిడ్స్ కానీ అదర్ ఫామ్ ఏదైనా కల్చరింగ్ అనేది జరగదు కదా సార్, న్యూ ప్లాంట్ ప్రొడ్యూస్ అవ్వదు కదా సార్.
రైట్. లెట్ మీ ఎక్స్ప్లెయిన్. For example, this is the plant, right? Actually, this plant unable to even it is produce the pollen grains or ovules. Due to some reason, it is unable to fertilize
and produce
and unable to formation zygote. As a result, no formation embryo.
Okay.
In this scenario, we selected the protoplast from this plant.
Okay.
This protoplast we selected nucleus. Either same or another plant. Only cytoplasm will require. Now this both fusion, protoplast and cytoplasm fusion. This is in vitro condition, not natural. But naturally, this is unable to form zygote. Unable to use this protoplast, some fuel gens, some electrode, some we fuse. We make it fuse together. As a result, cultures are formed. Now these cultures under tissue culture technique process, we get more plants. That's it. So mainly this is applicable where the plants unable to produce the seeds. Okay.
Like ornamental plants కోసం అయితే ఇస్తుంది సార్.
Maybe any type of plant, not only a particular ornamental. Clear? Can I leave the class?
Okay, thank
you. If you have any
sir.
doubt, ask me.
Okay, thank you, sir.
ఓన్లీ అన్నారు.
Thank you, sir.
Thank you, sir.
Just wait.
వేరే సెల్ నుంచి తీసుకున్నారు. సో ఇక్కడ న్యూక్లియస్ ఉంది, కానీ వేరే సెల్ లో న్యూక్లియస్ లేదు. సో అప్పుడు కూడా ఇది హ్యాప్లాయిడ్ అవుతుంది కదా, హ్యాప్లాయిడ్ అవుతుంది. ప్లస్ అగైన్ సేమ్ క్వాలిటీస్, నాట్ ప్రొడ్యూసింగ్ పాలన్ గ్రెయిన్స్, నాట్ ప్రొడక్షన్ ఆఫ్ ఎంబ్రియో, ఇవే క్యారెక్టర్స్ కంటిన్యూ అవుతాయి కదా సార్.
ఓకే, సేమ్ క్యారెక్టర్ కంటిన్యూ అవుతాయి, నో ప్రాబ్లం. ఇప్పుడు you selected the protoplast from the vegetative cell, not reproductive. What is the ploidy of vegetative cell?
See, N haploid.
Which I am asking about vegetative leaf.
Haploid only kada sir.
Diploid.
Leaf stem.
Just wait. Just wait.
Okay sir. Okay sir.
Diploid plant, right? First, just wait. Let me complete. Diploid plant.
Okay sir.
From the diploid plant, flower also diploid.
Okay sir. Okay.
Flower parts also diploid. That means androecium, gynoecium also diploid. From the androecium, group of stamens. Just right.
Okay.
Here ovules, right? Trains by meiotic. Here also by meiotic division. What is meiosis?
Reduction division, sir.
Reduction. That means this plant body is diploid. Of reduction division, it is haploid nature.
Haploid.
Okay. So, but you selected the protoplast from vegetative cell only, but not this. That is one.
Okay.
But another way also you can select this half. You can select the protoplast from the haploid cell also. In this scenario, instead of diploid plant, we get haploid plants only.
Okay.
Right? What if you selected the two different protoplasts, the two different plant protoplasts? Two different plant protoplasts means two different protoplasts ploidy 2n 2n. After fusion, what will happen?
4n.
4n tetraploid forms. So anyway, ploidy change. Characters also change. Okay?
Okay.
Clear?
Okay, sir. Okay.
Okay, sir.
Right.
Thank you.
और कुछ क्लास है क्या?
No.
Okay. Thank you.